Buongiorno, bentrovati, sono Giuseppina Nicchiarico del coordinamento regionale Malattie A.Ma.R.A.M. Buongiorno a voi, per me è il vero onore di essere qui, parte dalla consapevolezza che in questi anni il coordinamento ha condotto il proprio lavoro partendo dalle necessità dei pazienti. Mi riaggancio inevitabilmente a quello che è stato appena detto. Comprensione e sensibilità. Noi abbiamo lavorato perché comprensione e sensibilità fosse portato nei confronti di questi malati, ma che la conoscenza consolidasse la capacità del nostro sistema pubblico ad accogliere meglio e in maniera appropriata questi malati. La folla di oggi è bellissima perché è il risultato di quello in cui crediamo assolutamente, che è la forza dell'alleanza. Il coordinamento malattie rare, che nasce solo nel 2010 in un sistema sanitario pubblico organizzato dagli anni 70 per le malattie frequenti, in pochissimi anni ha puntato all'alleanza con l'università, che qui oggi vede i migliori rappresentanti nell'ambito e nella materia di cui ci stiamo occupando oggi, con gli ospedali, che sono un'alleanza importantissima, perché un bambino, e da lì che nasce un po' l'esperienza di oggi, un bambino che viene fatta la diagnosi, viene fornito un piano terapeutico in qualsiasi parte d'Italia e del mondo, deve riconoscere negli interlocutori del proprio territorio degli alleati. Uno dei bambini di A.Ma.R.A.M è riuscito ad effettuare, grazie al nostro intervento e all'organizzazione a nodi della rete malattie rare in Puglia, è riuscito a effettuare la propria terapia. È un bambino affetto da PANDAS che ha avuto la diagnosi a Roma, a cui è stato fatto il piano terapeutico a Roma, ma che ha avuto riscontro nell'ospedale qui dell'Alta. È inutile fare i nomi sono quelli di tutti voi. Oggi abbiamo anche qui a Gravina una persona che viene dall'altra parte del mondo, no? Perché quelli dei malati rari sono viaggi tante volte intercontinentali, e noi abbiamo imparato a far muovere i ricercatori, a far muovere gli scienziati. Abbiamo capito che dovevamo ragionare in termini di reti che affondano le proprie radici in questo bellissimo sud e in questa bellissima Puglia, per cui auguro la migliore prosecuzione dei lavori e darei la parola alla professoressa Cunningham, che viene dall'Europa. Prego.
Thank you so much. It is an honor to be here today. I have to take them off. Can't listen to the Italian and speak English. Thank you so much for inviting me. It is an honor to be here today and be in the presence of all of you who have spent so much time getting ready for this meeting. Also, many thanks to the wonderful group of mothers and people that have provided the impetus to bring to you today all of the speakers, including myself. So, can you hear me? Someone from the back, am I not speaking loud enough? Am I not speaking loud enough? Is that okay now? Am I loud enough? Yes. Wonderful. Okay. I, of course, bring greetings from the United States, and it's an honor and pleasure for me to be here in Italy with you. My correspondence was with Giuliana in Florence and also Dr. Falcini, who is here, and I certainly want to give a special thank you to them and to all of you who have taken such great care of me since my arrival in Bari. They even came to the airport with flowers. My goodness, I can't say thank you enough for all the special greetings that I had when I arrived. So now we need to talk about pandas. and the diseases of the streptococcus. And so the title of my presentation today is Autoimmunity, Microbes, and Neuropsychiatric Disorders. I have speaker disclosures because I've been very fortunate over the past almost 35 years to receive funding in the United States from the National Heart, Lung, and Blood Institute and also from the National Institute of Mental Health. Much of that has been for the study of rheumatic fever and the manifestations, and including PANDAS. The American Heart Association, American Diabetes Association, our own Oklahoma Center for the Advancement of Science and Technology, and many other groups, the International OCD Foundation, PepsiCo, the David Judah Fund from Harvard University gave us quite a lot of funding for the study of 1,000 patients with PANDAS in my laboratory. Autism Speaks has supported us, as well as the Autism Research Institute, and my financial affiliation is with Molecular Labs, where I'm co-founder of this company, and Weiss Lab in Sweden. So the Group A streptococcal diseases, you've already heard Dr. Savasta give his absolutely beautiful presentation of Sydenham, Korea. But the Group A streptococcal diseases, as you know, include sore throat or pharyngitis and skin infections, not to mention all the other very severe diseases such as cellulitis and necrotizing fasciitis, scarlet fever, septicemia, acute glomerulonephritis, and then our special disease of interest today, which is acute rheumatic fever. This may be related to PANDAS, as you've heard, and the major manifestations of rheumatic fever include polymigrating arthritis, carditis, which I've spent much of my life investigating, but today I'll talk about Sydenham, Korea, the major brain or neuropsychiatric manifestation, movement disorder that you've heard so much about from Dr. Savasta. Erythema marginatum and subcutaneous nodules are manifestations that are seen on the skin. The newest or latest revision of the Jones criteria is published in Circulation in 2013 by Jewett et al. So the pathogenesis of streptococcal sequelae, and probably all infection-related sequelae that come to us at the mucous membrane as we either inhale or contract disease from other people. The pathogenesis of the disease begins at the mucosa, and you can see this at the very top of the screen. I really apologize. I have beautiful colored slides, but the computer that I'm using actually removes all the color from my slides. So the point will still be the same, that the organisms bind to pharyngeal epithelium, and then, of course, the immune system in our bodies recognizes pathogens. And as it recognizes these pathogens, it makes an immune response to try to prevent illness in our bodies. And as this happens, we generate antibodies. Antibodies are in our blood, and they help prevent infection, and they also can turn on our body and cause autoimmunity, or autoimmune attack of organs such as our heart and our brain. And this is what happens in acute rheumatic fever, and you'll see at the lower part of the slide the tissue and organ-specific manifestations, including from the heart, carditis, in the brain, Sydenham, Korea, and then the joints or arthritis in the skin, of course, the other manifestations. But it's the immune response that is so intriguing about the diseases that are related to rheumatic fever. The definition of the term molecular mimicry is defined as the sharing of antigenic determinants between a host and the invading organism. And in the case of the streptococcus, it has the M protein and the group A carbohydrate on the surface of the bacteria that actually induce very strong immune responses. And as the immune response is made, then in some individuals that are susceptible, it attacks the heart and the joint or the brain, causing carditis, arthritis, or Sydenham-Chorea. Sydenham-Chorea and neuropsychiatric disease are associated with each other. In streptococcal-associated behavior and movement disorders, you see that there's Sydenham-Chorea, which you've heard much about, which is the mild incoordination to severe disruption of movements and emotional ability and other neuropsychiatric symptoms. However, PANDAS or PANS, pediatric autoimmune neurologic disorders associated with streptococci, or pediatric acute neurologic syndrome. These, of course, are related to obsessive compulsive behaviors and other neuropsychiatric symptoms, hyperactivity and emotional ability, and tic-like symptoms. We're skipping the video because I'm using your computer here, but you saw so many wonderful videos from Dr. Sebastian. So I'd like to go over for a few moments for those of you that may not be aware of just how associated Sydenham-Chorea is with neuropsychiatric conditions. First, Dr. Sydenham in the 17th and 18th century published on chorea and dramatic fever and exclaimed and reported that psychosis was a part of this disease in some individuals. And so it was a type of psychotic chorea that he reported. Among 100 patients with schizophrenia, 9% have rheumatic valvular heart disease, while the normal population incidence is 1.7%. So there is a great connection between rheumatic fever and schizophrenia. The retrospective study of well over 2,500 schizophrenic patients shows that there are 9 times more prevalent rheumatic fever, that the prevalence is 9 times higher in rheumatic fever than in the normal population. And there is 2 times more mental illness in children with rheumatic fever. 6.1% incidence of mental illness in 943 rheumatic fever and Sydenham-Chorea patients compared to 3.1% in non-rheumatic fever, non-Sydenham-Chorea children. Siblings of rheumatic fever patients had increased rates of functional psychiatric disorders, and then a high rate of histories of rheumatic chorea in psychiatric patients with psychoses are compared to other types of patients in the same hospital. So it's very clear that there's a very increased rate of mental illness and psychiatric disease amongst rheumatic fever itself. And there is no confusion about what rheumatic fever is. It comes from an infection. It induces an immune response that attacks our brain and our heart. One of the earlier stories about pandas was published in the Washington Post in 2011. And the story goes, a mother barely had time to react as her son lunged for the door of her car full of children, trying to wrench it open while yelling frantically, "I've got to get out." The mother managed to pull to the side of the winding road near their Charlottesville home as her son, nearly 8, leaped out of the car. He kept saying, "The car smells funny," and refused to get back into the car. She recalled, astonished that her normally self-possessed second grader would fall apart in front of his little sister and her friends. who stared goggle-eyed from the back seat. When the mother's efforts at reassurance failed, she called her husband, who left work. After an hour, the child's father managed to coax their son into his car, and they drove home. So this is really a mild incident. This is not a situation that many of you deal with children that are severely ill. But this is a similar situation where PANDAS begins with an incident and a child is perfectly healthy, and then suddenly overnight they become what a parent would call, as my son went crazy yesterday, suddenly. Criteria for PANDAS include the presence of obsessive compulsive disorder or tic disorder. Generally, it's prepubertal onset with an episodic course of symptom severity, acute, abrupt onset, and exacerbations, an association with neurological abnormalities, including choreiform movements. And I'd like to talk about this just for a moment and stand up and show you what I'm talking about. In Korea, you saw very large involuntary movements. In the case of the choreiform movements seen in PANDAS, the child will have piano-playing movements of the fingers, and also I can't do it with my toes for you. I don't have the video, but the toes also on the floor will go in a piano-playing manner. And so these are the small piano-playing choreiform movements that are often seen in cases of PANDAS. And this is the acute onset form of PANDAS. Actually, it's important to clarify that because so many children become chronic. So the acute phase or the acute form of PANDAS, many times often seen are these choreiform movements. A temporal relationship between symptom exacerbations and streptococcal infections can be seen. We know now that other infections may also play a role. The clinical manifestations are there can be a relapsing, remitting symptom course, as many of you as mothers of these children have seen. The young age and onset is generally about 6 years old for tics and 7 years old for the obsessive compulsive disorder. Boys outnumber girls 3 to 1, and comorbid tics and OCD are common, about 65%. And we've shown in our publications that tics and OCD are most common in streptococcal infections, so you will see both of them in 65% of the cases if they have strep. Other comorbid symptoms occur frequently. And of course, as Dr. Sebasta pointed out, there are many of these other symptoms. And you can see the comorbid diagnoses that you might find: ADHD, ODD, depression, dysthymia, separation anxiety, over-anxious, enuresis, and anorexia. Symptoms during exacerbations, as I said, in the acute phase or acute PANDAS, which is what was discovered first. The first 50 cases that were discovered by Dr. Susan Sweeto at the National Institute of Mental Health all had these choreiform movements, and they are part of the acute onset. You generally would not see them in the more chronic cases as they progress beyond a year. Emotional mobility, school changes, personality changes, bedtime fears, fidgetiness, separation fears and anxiety is certainly one of the most important, where a child will be afraid even at 12 years old for their parent to even leave them alone. sensory defensiveness, irritability, impulsivity, and distraction. So oftentimes, as you saw from Dr. Sebastia's talk, he discussed the writing abnormalities because in Korea, the problem is that the milk-maid grip, they can't hold onto anything. And this actually, you'll see later in my talk, we have an animal model that actually does the same thing. But in a case where they were drawing perfectly, once they had pandas, they couldn't draw anything at all. And this is very similar to what happens also in Sydenham, Korea. So the model of etiology for the pathogenesis of pandas is very similar to the pathogenesis of those sequelae seen in rheumatic fever. You can see that there would be strep bacteria that children pass between themselves frequently, genetic susceptibility in the host, an abnormal immune response, and then the result is getting pandas with OCD and tics. One thing that we have learned is that antibiotics prevent rheumatic fever. In the case of pandas, and it's been a lot of controversy about what to do for antibiotic treatment in pandas, but it's becoming very clear that antibiotics do help control the symptoms of pandas, because when antibiotics are given during an episode, if they're rapidly removed, then the symptoms return, and sometimes even more viciously. So it's important to understand that antibiotics are important in pandas, especially with the strep. Other infections and other antibiotics might need to be used. So that's one place, and antibiotic prophylaxis is easy compared to immunomodulatory therapies, where if the disease progresses on, to the abnormal immune response that is uncontrollable, where it can't be stopped if the infection is stopped, then immunotherapy is required, and that's much more complicated in children. The immunotherapies are intravenous immunoglobulin and plasmapheresis or plasma exchange. And then also in the United States, they are using rituximab to destroy the B cells that make the antibodies in some patients that have life-threatening disease. And also the group at Stanford currently now is using antibiotics and steroids, and I know that Dr. Sebastia discussed that also. It's very, very effective, antibiotics and steroids together, and also antibiotics and IVIG together if one has to utilize IVIG. The MRI evidence of regional inflammation is shown in this slide of the MRI, where you can see the enlargement of the caudate. The caudate and putamen are the regions of the brain that are affected in Sydenham, Korea, and also in pandas and pans, and the globus pallidus, but not in the thalamus. Change in OCD severity 1 month following treatment with IVIG, placebo, or plasma exchange. This was the first IVIG trial and plasma exchange trial that the National Institute of Mental Health had in the early, I would say early, maybe 2000 or late 1990s. The reference is to Perlmutter et al., showing how effective IVIG and the plasma exchange are. The plasma exchange is slightly better than the IVIG, and you can see that the placebo group did not change in their behaviors as recorded by the Yale behavior obsessive compulsive test, the Y-box test. So just to talk a little bit about the approach to the study of Sydenham chorea and related disorders. What we did, I'm going to share our research over the past nearly 20 years with you about how we began to understand the role of antibodies in Sydenham chorea and also in pandas. We had developed technology in our laboratory for producing monoclonal antibodies from an individual who was ill. We took the blood and made these antibodies and studied the brain for its autoantigens and streptococcal antigens, and then studied the pathogenesis of the disease. The reactivity of Sydenham chorea-derived monoclonal antibodies was first identified with the antigen lysoganglioside GM1, and you can see these were monoclonal antibodies produced from a Sydenham chorea patient that I saw myself, and we obtained their blood and then produced the monoclonal antibodies. And you can see that they all react with the lysoganglioside GM1, whereas our control antibody does not. The antibodies were highly specific for lysoganglioside and another monocelluloganglioside, whereas the other gangliosides that are so evidently recognized in Guillain-Barré syndrome are not recognized in Sydenham chorea. It's another type of reactivity. So the chorea sera, once you look at a monoclonal antibody, it's almost only a single antibody you have made as a reagent from a patient. So then you have to go to the serum of the patients. And so the NIH provided us with patients with Sydenham chorea, and the chorea sera were all very reactive with the lysoganglioside. So our monoclonal antibodies could be used as reagents to study the pathogenesis of the disease. And you can see that in convalescence, the anti-lysoganglioside antibodies dissipated. So what about the anti-lysoganglioside antibody in pandas? Well, we have done studies with the pandas children from the NIMH, and this is a small group, but we've looked at many, many others since then. And you can see that it's highly significant. The titers of lysoganglioside are highly significant in pandas and in Sydenham chorea, and also in Tourette's syndrome, which is a chronic tic disorder. However, ADHD in normal patients did not show elevations. Or we use all of the significant levels are compared to normal. And then we ask the question, are these antibodies in the cerebrospinal fluid? And of course, this is a very important question because how do antibodies get into the brain? They get into the brain from the cerebrospinal fluid, or CSF. So when we stain the CSF, and I'm sorry you can't see it here, but the slides are pink, where the CSF stains from Sydenham chorea sera and from pandas, and the controls are all negative. I could turn my computer around, but I can't. The colors are all washed out from the computer because I have a Macintosh and they have a PC. And we tried to put my computer up, but it didn't work with the equipment. So we're having to show what I have here. I'm glad to show you on my computer after my presentation. So any that have questions, the slides for Sydenham chorea and pandas are very red, whereas the controls are very blue. So the anti-tubulin antibody is also important. Tubulin is actually enriched in the brain because inside the cells of the brain is tubulin. just like inside cells of the heart is myosin. Tubulin is recognized by these antibodies that saw lysoganglioside. That's what molecular mimicry is about. And so the tubulin antibodies were significantly elevated in pandas and also Sydenham, Korea. And notice they were pretty elevated in Tourette's syndrome also, mainly with the ticks. Notice that they did not track with the OCD. And so it's a very good antibody that we see elevated in ticks. So again, the antibodies had recognized lysoganglioside on the surface of the cell and tubulin is an intracellular molecule in the brain. However, we wanted to look and see if it might bind to the outside of living neuronal cells. And so that's what happened. You can see the little lumps. They're not stained bright red like they are on my computer, but you can see where they've stained the outer part of the neuronal cell. This is the Sydenham Korea monoclonal antibody. These dark regions are staining with the antibody, and then a commercial anti-ganglioside antibody does the same thing. You can see the lumps on the outside of the cell by the antibody. I'm just sorry you can't see the red. And then the control is blue. So as far as Sydenham, Korea, we felt that there was a very big case for mimicry between the group A carbohydrate epitope of the streptococcus, which these antibodies recognize, and lysoganglioside and tubulin. And we asked the question, since we demonstrated that the antibodies bind to the surface of a neuronal cell, then how can they cause disease? What happens to that cell? Do these antibodies kill the cell? No, they don't kill the cell. I'm not showing you that data, but we tested for that. They don't kill the cell. What they do is they bind and they trigger an enzyme called calcium calmodulin-dependent protein kinase 2, or CAM kinase 2, in neuronal cells. They bind to something on the surface. Maybe it's the lysoganglioside, or I will talk about dopamine receptors later in my talk. Maybe it's the dopamine receptors, or maybe it's both of them. So what we did was we looked at our monoclonal antibodies, and we saw that one of our antibodies from our patient with Sydenham, Korea triggered this enzyme. And I always think, oh my goodness, it gives me chills to think, what if we had not had that one antibody? The other two are great controls because they are more or less negative. They don't signal as well as 24. And so we've used 24, that monoclonal antibody, to study the mechanisms of disease. We also wanted to show that that monoclonal antibody is not the only, is in the patient's serum. And so you can see that the patient's, the donor serum, triggers the CAM kinase in neuronal cells, whereas when the patient became well, and they actually were treated just with, bed rest, to be quite honest, and they got better, and after they became well, they had no antibody. So there is a great correlation between the presence of the antibody and the symptoms of Sydenham, Korea. As you can see here, 5 more patients from Dr. Sweeto at the NIMH, and then the convalescent patients where in blue there is no activity. So what is the mechanism? These antibodies bind to the neuronal cell and cause excess amounts of dopamine to be produced. And you can see in these studies of the Korea monoclonal antibody and the Korea serum from patients at the NIMH that they trigger the release of tritiated dopamine from neuronal cells. So that's what's happening. It's in the brain, in the region where the antibodies are binding to neuronal cells. It is causing the cells to make too much dopamine. We looked at pandas, and because we thought, well, pandas' antibodies are really similar, if not almost identical to the ones that we have investigated in Sydenham, Korea. And so what we found was the pandas patients, when compared to Sydenham, Korea, were less strong. They were not as strong in signaling the enzyme, but they clearly signaled the enzyme, the CAM kinase 2, similar to those of Sydenham, Korea, whereas another non-pandas group did not produce this type of strong signaling. So the idea is that the antibody binds to the neuronal cell and it triggers CAM kinase through tyrosine hydroxylase, which is the identifier of a dopaminergic neuron. So already we're starting to think about dopamine receptor. But the release of excess dopamine is likely part of the cause of the movement disorder. And I know you can't see this slide, but what we did was we actually took the monoclonal antibody and we put it into the brain of a rat. And when we put it into the brain of a rat, it actually triggered the tyrosine hydroxylase. And that meant that the antibody, once it got into the brain, could actually trigger tyrosine hydroxylase in living neurons in a living animal. And so we didn't want to stop there, however. We wanted to prove that if we made our antibody into one that actually could penetrate the brain, that we would figure out where it went once the immune system made the antibody, because we made it in a transgenic animal, and so that animal's B cells made the same Korea antibody, and it actually went to the brain. Oh, actually my picture looks good. So you can see the basal ganglia. And actually, I had experts look at our slides, and the staining is either in the substantia nigra or in the ventral tegmental area. It was too difficult to be able to discern which one. But you can see with the green staining, it does take away all the red, which of course indicates the staining of the dopaminergic neurons by the anti-tyrosine hydroxylase. So these would be red, but in the staining, this is where the antibodies targeted in the basal ganglia. They are not in the hippocampus. There are a few that are stained in the cortex. But this slide was also supposed to show that they were dopaminergic neurons, but you can't see that because the red has been removed from the slide. But I can guarantee you that they are dopaminergic neurons. So is there evidence for a role for dopamine receptors in Sydenham Korean pandas? And we propose the hypothesis that dopamine receptors present on the surface of neuronal cells would react and respond to the antibodies as if they were neurotransmitters such as dopamine, so that the antibodies actually would mimic dopamine. So the answer is yes, and we show this where the monoclonal antibody first was shown to signal the dopamine D2 receptor that's expressed on a cell. We have transfected cells that are transfected with the gene for the dopamine receptor. And so you can see here the signaling of the D2 receptor, whereas in controls, no, and then a very good dose-response curve where we use different concentrations of the antibody. And that shows that the antibody very clearly signals the dopamine D2 receptor. Well, we can't be happy just with investigating the monoclonal antibody. We must also look at Sydenham Korea sera. So this also signals, and it's an inhibitory. That's why it goes down. is the signaling of D2 is always inhibitory. And so that's why in this test, the sera causes the reduction in the cyclic AMP. So what about PANDAS sera? PANDAS sera also does the same thing as the Sydenham-Korea sera. You can see the PANDAS sera signaling and causing the decrease in cyclic AMP levels. So yes, PANDAS sera also had the D2 receptor signaling antibody. And so next, of course, then we wanted to find out, do the antibodies react with the D1 receptor, which is the activating receptor? Remember I said the D2 is inhibitory and D1 is activating. So imagine if you have antibodies to both of them, you've got one saying, oh, inhibit, the other one saying, oh, activate, and it can cause, obviously, we believe, havoc in the brain. So we did the same thing with PANDAS cases. We made monoclonal antibodies from cases and looked, and sure enough, we found antibodies that react with the D1 receptor. And then this just indicates peptides from the D1 receptor. These peptides react with the B4C, which is the monoclonal antibody, from a PANDAS case, actually from the NIMH IVIG trial that was recently published. This simply shows the peptides, and the D1 receptor is D1E2. This is where the antibody binds to the receptor. And if we use the peptides in the patients from the trial, we show that the peptides will also be recognized by the patients significantly. And so what would a D1 receptor autoantibody do in this disease? And so when we mix the antibody with dopamine, we found that the activity increases about 50 times what it would be with just the antibody alone. So what happens is we can predict an antibody to the D1 receptor would change the appearance of the receptor so that it becomes much more active. And this will end up and potentiate the effects of dopamine, make dopamine much more efficient, 50 times more efficient on receptors. So you've got too much dopamine being produced if you're binding the D2 receptor, and then you're turning around and making the receptors that recognize dopamine much more sensitive to dopamine. So you can understand how a person might go crazy if they had all of this going on in their neurons. And so we did look clinically to see if these antibodies are present in the direct ELISA. And you can see for the D2 receptor, they're highly elevated in PANDAS and also in Sydenham-Korea. And the anti-D2R antibodies are present with the little choreiform movements. In those patients, they're very clearly there. They're in Sydenham-Korea almost 100%, and seem to go in PANDAS with these fine choreiform movements in acute cases of PANDAS. The anti-D1 receptor antibodies seem to be elevated primarily in the OCD and also in PANDAS. And in Sydenham-Korea didn't become significant, but they are highly elevated because they were so spread apart that we didn't get a good p-value. So we have publications that show evidence for autoantibodies against the streptococcus in brain in both Sydenham-Korea and in PANDAS. And these are listed on the slide. From our first publication in Nature Medicine that shows mimicry and autoantibody neuronal cell signaling in Sydenham-Korea, to the autoantibody neuronal cell signaling in PANDAS, and that's in the Journal of Neurology. Then there's a review, but this third paper shows the tritiated dopamine experiment and the release of dopamine from neuronal cells in large quantities in that paper. And then the tubulin paper is in the Journal of Immunology. And then there's two other papers that I will be talking about the data shortly. The Ben-Pazzi paper in PLOS ONE is about the dopamine receptor autoantibodies, and then another one on the transgenic animal, but I've already spoken about that. There are also a couple of publications you should know about, because PANDAS is not just acute. PANDAS is chronic. And just like Tourette's is chronic, Tourette's definition is that tics continue for greater than a year. And we have two studies, this is new, they're not as new as they were when I made the slide, where we talk about the anti-neuronal antibodies in a heterogeneous group of youth and young adults. That was from the 1,000 patients that we studied in Oklahoma. And then the Singer paper is about a group of children that Dr. Harvey Singer, in a collaboration, he had 8 children that we followed over a couple of years of time, and showed that the antibodies in chronic cases sometimes stay elevated. They never go away. And so when you have an infection or a traumatic event and are able to potentially break the blood-brain barrier, then these antibodies may enter the brain. We have animal models. Not only did we study Sydenham Korean pandas in humans, we wanted to have an animal model because it's good to have an animal model that you can show does the same thing as in humans. Oh, and I need to say that the animal model was developed with me, but also they developed it at Tel Aviv University in Israel. Dr. Daphne Joel, Dr. Leor Grinberg, it was actually her thesis, and Dr. Daphne Lotan, who continued to work with us over the years in this Lewis rat model of streptococcal-induced behavioral changes. So after giving animals streptococcal immunization, the behaviors are affected. Just like in Korea, they can't hold a pencil, well, the animals can't hold their food. And you can see the food manipulation goes down. And that they can't walk a beam because they can't hold a narrow beam, they can't hold onto, and that's shown in the difference between the narrow and wide beam. And then as far as OCD, when you spray the animals with water, they groom for hours, whereas normal animals stop grooming themselves after they've removed the water. And then the activity curve is not significantly different. And the symptoms in these animals were relieved when they were treated. These are very complicated slides, and I know you all probably want your lunch, so I won't spend hours on the slides, but haloperidol and also paroxetine change the behavior back to normal. These are treatments. The paroxetine is a serotonin drug, and then the haloperidol blocks the dopamine D2 receptor, so it makes sense that it would work. The antibodies were tested from the blood of the animals, because if they had the behaviors, then we want to know, do the animals have the same antibodies that the humans do? And the answer is yes, they do. They have the signaling of the CAM kinase, and this is removed. If you use anti-IgG, you remove all the signaling. In other words, when you remove the immunoglobulin from the serum, you remove the signaling. So it is antibody that's causing the signaling. If you look at the dopamine D1 and D2 receptors, you actually find that they are positive. from the sera, and we used Western blots to make sure they were the right size molecules and also signaling. I don't know what this is. Thank you. Okay, we'll just come back. Yeah. I'm so thankful for help. So now that we have shown that the animals get, if you give them strep, they get behaviors, and if you give them strep, they get the antibodies that actually are similar to the human Sydenham-Korea pandas. So then we look at the brain, and does the brain look like our transgenic animal that we gave the human antibody to? What happens when we give the strep immunization to the Lewis rat? We get antibodies, oh dear, that react with the striatum, and the staining is yellow, so you'll just have to pretend that it's dark brown. It reacts with the striatum and with the cortex, and somewhat with the thalamus, but not as much. And you do not see reactivity with the hippocampus or the cerebellum, very similar to what we got in the transgenic animal, where the antibody was actually naturally produced and was directed to the striatum in the brain. Whereas here, we immunized and the antibody was directed to the striatum in the brain. So it's just a lot of evidence, and ours is not the only evidence. There are other animal models that were devised by the Hornig lab. Hoffman et al. In 2004 saw a jumping movement, and they took it as a video where the mouse jumped and jumped and jumped after being immunized with strep. And then another where they passively transferred the antibodies, and we did that too. When you passively transfer the antibodies, you get the behavior again. So the antibody, you purify the immunoglobulin from the serum of the animals and give it back to the animals. And when you do that, they develop the behaviors again. So then the question, is Sydenham-Korea a streptococcal-induced autoimmune sequelae? Is it a path to understanding neuropsychiatric disorders associated with autoimmunity and infection? Because I actually feel that this is not just associated with pandas. This is well beyond pandas. It's that infections and inflammation affect your brain in all the time. We're always having effects in individuals that have neuropsychiatric disorders, whether they're adults or children. We just are at the point in our research where we're beginning to understand what's happening in the brain and how much inflammation plays as a role in leading to behaviors that are altered. So Dr. Hilleman Pazzi did a study of another group of Sydenham-Korea children in Jerusalem at the Shari Zedek Medical Center there. And we were delighted to find the same results that we saw with the NIMH group, where the D1 and the D2 antibodies were elevated. And you can see this, that both have good p-values. And the ratio actually is associated with, correlated with disease. I'll show you that in a moment. But one of the important things that we've learned from this study is that the D1 antibody comes up first with the neuropsychiatric symptoms. And so that's probably why it seems more prevalent with OCD. And then the D2 antibodies seem to elevate later with the movements that you actually see. Parents of children that have Sydenham-Korea probably didn't notice the neuropsychiatric symptoms so much as suddenly their child became so violently having involuntary movements that it just simply overrode the idea that they also had a neuropsychiatric problem. And this shows that the dopamine receptor antibody ratio correlates with neuropsychiatric symptoms. And recently we published a review in Nature Reviews, and they drew this lovely diagram of how the antibodies, the molecular and cellular basis of Sydenham chorea, and I would say also most likely of pandas. And you can see, except you can't see it, it's so sad, that the antibodies bind to the neuron and release large amounts of dopamine, because actually we can't see any of it. I'm sorry, there's no point in even. This is better. This is much better. This is the model of autoantibody effects in the brain, binding of high-affinity cross-reactive antineuronal antibodies to lysoganglioside and dopamine receptors may cause alterations in dopamine neurotransmission. You can see both the neuron and both the postsynaptic and presynaptic situation where too much dopamine is being released and the postsynaptic neuron is responding. So that autoantibody-mediated increases in central dopamine levels and selective activation of the D2 and D1 receptors combine to produce altered movement and neuropsychiatric symptoms of these diseases. So how could these diseases occur? What are some of the ideas that we might have? Well, as I've told you, there will be antibodies leading to excess dopamine. But also in people that are susceptible, what are their possible genes that could be mutated or could actually lead to this idea? Maybe there's a failure to degrade the dopamine. So then if you have a whole other set of problems where you make too much dopamine, and then what if you can't clear the dopamine? Your enzyme to degrade the dopamine is not good. This is a possibility, and there are enzymes that are important in. It's actually one of the methylase enzymes, I believe. It's gone from my slide. Oh, here it is. It's a catechol. Here it is. Catechol O-methyltransferase is important in schizophrenia, and they believe it's because that the patient cannot degrade dopamine. And it's possible that in our patients with Sydenham chorea and also with pandas or PANS or any of these neuropsychiatric symptoms from infections, that one of the problems is that they have other things, other factors that are important. And so the concept of autoantibodies and autoimmune diseases is not a new one. But the concept of autoantibodies in mental illness is new. And so it's always difficult for people to accept new things. The status quo is what people love, and the change is what people fear. And so the fear of changing your idea, even in the face of data, which it would be hard for me as a scientist to say that this data doesn't show that this exists. And so the idea of pandas may have originally been controversial, but over the last nearly 20 years, there has been research that has shown that the antibodies do occur and molecular mimicry does occur, and that there are groups of autoantibodies against different antigens that are associated with a particular type of autoimmune disease. In our case, the antineuronal antibodies have greater than 91% sensitivity for the disease. If all 5 of these things are tested, the 4 ELISA antigens and the CAM kinase, it's almost 100% for sure that a child with pandas or synhapperia will have one of these. In pandas, PANS, tics, and OCD are some neuropsychiatric diseases associated with inflammation. Anti-neuronal autoantibody groups against certain neuronal antigens and receptors are a characteristic feature. And then one other area where we could say that things might go wrong and your child or an adult might get disease from autoimmunity is immune complexes that form must also be degraded. If the genes to degrade the immune complexes are not available in a person, this may also be another set of genes that are affected in persons with autoimmune pandas or chorea. So I'm almost finished with my talk, but I added 3 slides last night that I wanted to show you today because they are new and I'm getting ready to publish them. And there's no reason not to show them to you. So the autoantibodies that are elevated in PAN study subjects from the most recent IVIG trial at the NIMH, you can see that and this is all the patients together. The placebo was treated, many of them had antibiotics, everybody improved. So all those pandas patients, as they improved, no matter what they improved from after they were treated, the antibodies decreased. And so it once again shows that the antibodies decrease significantly after treatment. And also that the symptoms are decreased when the antibodies decrease. I'm not going to go through every single one, but again, antibodies decrease in recovery and improvement. This is another set of patients from my own group, and you can see how the antibodies decrease, decrease, decrease. It all happens as these people are improved or recovered, the antibodies go away. However, in people that get worse or that don't recover, the antibodies don't go away. Remember, I told you in chronic disease, the antibodies generally don't go away. It is a problem if it goes on chronically, that the antibodies remain either the same or they even, as you worsen, when you have a worsening episode, the antibodies actually go up in patients as they worsen. And just, you probably can't see it, just to remind you that if CSF is investigated for the antibodies, they are there. They are detected in the CSF. And the disease symptoms then may be a 2-step process. One is the immune response against the infectious pathogens, such as the antibody response. And I'm not saying innate immunity doesn't play a role. The microglia are very important because they will recognize the immune complexes of these antigens with their antibody when they are not cleared. And that is very important. What happens then is the blood-brain barrier must open and allow infection, or by infection or stress, to allow the antibodies to penetrate the brain. So there are 2 ways you can break the blood-brain barrier, with molecules from infection or with molecules that are created by stress. Those are 2 very important factors that will break the blood-brain barrier. And so that ends my presentation, but I'd like to thank many people, my whole laboratory that has helped me all these years, particularly Dr. Christine Kerman, who helped me initially in our study. She was a postdoctoral fellow in my laboratory and came to us from California. I always love it when they come from California to Oklahoma because people love to stay in California, and I'd like to see them come to the center part of the U.S. But group at the National Institutes of Mental Health. We could not have done all this work without Dr. Sweeto, who called me when she first discovered pandas and said, would you please help us? And of course I said yes, and we have all of this data from her patients. Yale University, Dr. Jim Weckman provided me with all of the Tourette's patients. And then the Tel Aviv University group, the animal model, and the Shari Zedek Medical University group, Dr. Ben Pazzi, the other SIDPAM study. And then I have collaborations with Dr. Singer at Hopkins and people at Harvard and at the University of Minnesota, Dr. Kaplan in infectious diseases. So for those people, we could not have done our work without the help of our collaborators. And certainly thank you again for your attention.
Ringrazio tantissimo la professoressa Cunningham che ci ha fatto vedere come attraverso proprio la ricerca scientifica, quindi studi approfonditi delle basi molecolari di queste malattie, insomma, abbastanza, un po' correlate tra di loro con questi sintomi neuropsichiatrici. Solo attraverso la ricerca si arriva poi all'identificazione di nuove terapie che possono essere efficaci. Quindi apro la discussione, giusto una domanda per la professoressa oppure per gli interventi precedenti.
Vorrei chiedere, la ringrazio per la relazione e lo spettacolare esempio di ricerca e innovazione e di contatto internazionale. Vorrei sapere se questi bambini avevano in uso antipsicotici.
The question is about antipsychotic drugs. The children that were studied did not have antipsychotic drugs in studies that I have, but children are often given neuropsychotic drugs initially because oftentimes their disease is misunderstood and not thought to be related to the immune system and is not thought of as an autoimmune encephalitis because probably the disease itself, even though a person may not be in a coma or be completely incapacitated, it still may be a mild encephalitis in the brain. So giving psychotic drugs may actually worsen the disease, but with that said, sometimes psychotic drugs do improve the behavior. So it is something that is a trial and error. And I know for Dr. Tanya Murphy, if she were asked that question, she would say that most of the time, the psychotic drugs in children with pandas are given a dose that is too large. The dose must be very, very small. It must be started at a very tiny amount compared to what the normal dosage would be. And then you can advance the dosage as you determine if the antipsychotic drug will actually work.
Ci sono altre domande?
Sì. Volevo chiedere, questa malattia l'ha definita una malattia nuova, per cui è ritenuta da lei nuova perché non era diagnosticabile prima, essendoci una clinica, oppure è una malattia che si sta verificando relativamente negli ultimi anni? E se è così, magari quali potrebbero essere le cause principali che scatenano questa patologia? Grazie.
question. That's a big question. There are no perfect answers. However, I believe that over the years in the 1940s and 1920s, before penicillin was used to treat rheumatic fever, that many children that had carditis actually died. They never had children. This is one answer. I'm going to give you several answers. The children that were so ill with carditis either could not bear children because they were too sick or they died. So the genes for severe rheumatic fever may have not completely disappeared, but many of them may have exchanged in our population such that we may see other types of diseases that are related to streptococcal infection or to other infections because of the genes in our population. On the other hand, Dr. Sydenham recognized the neuropsychiatric behaviors in rheumatic fever, and he even called a rheumatic psychosis. So it is possible, and I think that it probably was evident in Sydenham's day, but it may be more prevalent now because the genes in our population have changed. I mean, since the discovery of penicillin, or before, when you couldn't use penicillin and children were gone. So it's one way or the other or both.
Posso fare una domanda? Allora, senti, io volevo chiedere se lei ancora crede delle malattie autoimmuni, dell'esistenza delle malattie autoimmuni, perché nel 2015 due università hanno scoperto il sistema linfatico del cervello a livello di sistema nervoso centrale, che ove i linfociti B e T non funzionano da delinquenti che peggiorano, diciamo, lo stato mielinico o neurologico del cervello, ma funzionano da operatori ecologici, cioè non fanno altro che pulire i danni che ha subito il cervello e ritardare la malattia. Ora, tutto questo, dico, io chiedo a voi tutti, sulle malattie rare e autoimmuni, avete mai prescritto un mineralogramma? Ove a livello tissutale noi rileviamo minerali e oligoelementi e anche metalli pesanti. Ora, il dottor Carrel, Alexis Carrel, nel 1912 ha avuto il premio Nobel, ma non per quello che dirò, ma per quello che ha fatto successivamente. Sì, no, 2 secondi, ho finito. Se diciamo, prima di fare tutte queste prove che voi fate, di analizzare, di far analizzare il mineralogramma, tramite il mineralogramma, tutti i minerali e oligoelementi, e verificare le reazioni su tutte le prove scientifiche che voi fate, perché gli oligoelementi sono...
Scusi, la domanda, se no non
sì, vabbè, altrimenti non capisce...
Ma non possiamo fare un'altra relazione, siamo già in ritardo, grazie.
Allora, la domanda è questa: secondo voi che funzione hanno gli oligoelementi minerali a livello di ioni su tutte le patologie? Grazie.
Well, of course I think that minerals are very important, and that oftentimes there are many things we don't understand. For example, the lymphatic system was not clearly understood of how it actually came into the brain until the most recent work was done, and that was very exciting. But that also does not negate everything we know about autoimmunity, because autoimmunity oftentimes begins with the immune complex that actually binds to the monocyte, and the monocyte actually spews or the monocyte in the blood actually makes cytokines, and that's very important. But it doesn't mean that the minerals don't play a role in disease. I mean, they're different things. And one of the problems with disease is that we can only in research, unfortunately, focus on one thing, because you have to have controls, and you can spend your whole life looking at a few questions, and then you realize, oh well, there's something else over in another area that is very important. And so I certainly think that it's very important for us to consider everything. My mind certainly would be open to any of the mineral effects on the brain. They are small molecules, and they're likely very important. And I'd like, since you asked me this question, I would like to say one other thing that I think is going to change all of our lives very soon in very drastic ways, is called the microbiome. The microbiome is very important in many diseases. In fact, I just recently copied a paper about Parkinson's disease and the fatty acids. Now, no, this is not minerals, but it's the same situation where there may be minerals that we don't understand. And we don't really totally understand the biofilms and the bacteria that are in our gut. And I would have never thought in my wildest dreams that fatty acids could have led to Parkinson's disease. Now let me give you a scenario of how it might apply, and then I'm going to be done. But it's very important because this may make you realize just how complicated autoimmunity can be. Is that the microbiome leading to the fatty acids. Phosphocholine, for example, has been published to actually stabilize membranes. Maybe if something like phosphocholine, or we understood what could stabilize membranes of neurons, maybe that is a potential problem. Antibodies bind, and then the neurons may be more sensitive to the antibodies because of the microbiome that you may carry in your intestinal tract. This is just an example. So just to leave you with some of those facts, that not only minerals, but microbiome, many other factors that we may not know about. We have to have an open mind. But the most important thing of all is to try to help the children and not argue about the cause. It's just to try to work together and do the best we can to come up with solutions of treatment that will actually help us understand the brain.
Ringraziamo ancora la professoressa Carica per la bella relazione. Interrompiamo adesso i lavori per il lunch. Riprendiamo fra 20 minuti al massimo. Grazie.
Prima di scendere giù consegnate le cuffie e poi scendiamo giù. Grazie.